Here's Gwen.
Well, thank you very much. My name is Gwen Hummel. I am a creation scientist. And again, if you guys have any questions, this is a 6-week course. So if you forgot to ask one today, I'll be here next week and the week after that, and we can get all your questions answered. So today we're talking about genetics and the Bible. And I wanted to ask you guys, I use a lot of blue. If anyone's colorblind or has any issues with that, just let me know if you can't see anything I put up on the screen. So, figure out. So I wanted to start today by just asking, what is the genome? Have you guys ever heard what the term genome? Do you know what it means? No? Okay, so we'll talk about what that is. So the genome is the complete set of genes within a cell or organism, and an organism is us, so any living thing. So the complete set of genes within those things, that's called a genome. And we're going to think about the genome in terms of books and literature. So if we're thinking of it as a book of life with letters, then that would be our nucleotides. So if you guys have seen the A, T, C, and G, that's our nucleotides, all the letters that make us what we are. And these are actually made up of chemicals, which we won't get into in this class. But when you look at this book of life, they're made up of all these letters. And when we put those letters together, they form words, and those words are called genes. So this is how we start building the information that actually puts us together. And then when those genes come together, they form books in a genome library, and these are our chromosomes, which that's a picture of chromosomes on this slide. And our chromosomes are little X-shaped packages that fill up all of our cells, that hold all of our information, and they make us who we are. And then these chromosomes, they form an entire library, and this is what the genome is. So we're going to talk about genetics in terms of these letters to words to books to a library. And this is a very simple way to look at genes without having to get too technical. So if you guys have any questions about this as we go forward, just let me know, but otherwise it's all on this slide, which will be posted later. So when we look at the genome as a book, we have to remember it's not exactly like a book. So it's dynamic, meaning that it's always doing things. Our genome's constantly moving and changing, because it's also self-regulating, meaning that in time, certain things are happening, such as we're breathing, we're taking in oxygen, and that has to be metabolized in our body. So it's self-regulating, making sure we don't die from poisons in the air, we're able to metabolize all the things around us. That's what that means. It's also multidimensional, because as I mentioned, this also acts within time. So remember, our genes when we were little told us to be little and we needed to grow and do all these different things. And now we don't have to grow as much. We're eating things differently, and so our genes are actually telling our body to do different things in different times. So remember, our genes aren't exactly like a book. They're dynamic, self-regulating, and multidimensional. And we're going to talk about this thing. It's called Neo-Darwinian theory. Neo meaning new, so this is modern, not from the 1800s when Darwin first wrote his theory. This is how most evolutionists think about genomes. So when we look at genomes, we're thinking of them as instruction manuals, remember our libraries and our books. Darwin says that these arose from a series of simple letters through a long series of mutations, and we can think of mutations as typos, so something that was copying along and messed up. And then he was also talking about natural selection, and that we can kind of think of that as copy and paste. So on our computers, we copy our sequences exactly, and our mutations are typos in that sequence. So Neo-Darwinian theory, the way modern geneticists think about genetics, is they look at these instruction manuals are built up of typos and just random copy and paste. But we have to remember, natural selection is blind and purposeless. It's a process where some things reproduce more than others for no real reason. So natural selection, many people attribute intelligence to natural selection. They say nature chose to do this or evolution was smart to do this. But we have to remember that natural selection has an IQ of 0. So it's not omnipotent, it doesn't have a plan, and we have to keep that in mind when we look at genetics and how things are acted upon in nature, because we don't actually see natural selection choosing a certain path. So, evolutionary theory, one of the biggest things is this word called abiogenesis. Has anyone ever heard that word? It's a pretty big word. Probably not. So abiogenesis, when we're talking about the genome, it's the theory that our nucleotides, so remember those are our letters, they formed from nothing and they came together to create a sequence also from nothing. So abiogenesis means that something generated itself from nothing. And in the genome, we have to remember that all of those little pieces, so our letters, our words, our books, and our library, had to have all come together from nothing. So some of the biggest problems, slide got a little messed up here, but some of the biggest problems are that the generation of amino acids actually creates toxins, toxins like cyanide and carbon monoxide, and this actually kills the amino acids if we're building them in nothing. So evolutionists believe that amino acids started out in water. This is an early Earth model, but amino acids actually are held together by hydrolytic bonds, and those means hydrolytic is hydro is water, lytic means it breaks. So water actually breaks these amino acid bonds, so we're not able to put them together in a natural environment. So these are two of just the biggest problems we have with the evolutionary theory of where the genome even came from. But we see that in nature, our amino acids create toxins, so it would kill the amino acids and we never get anywhere. And they create hydrolytic bonds, so the water that they're supposed to have originated in would actually prevent them from coming together at all. So here's a big question. Are random mutations good? Remember, when we're looking at the genome, we're looking at evolution, they say everything came together because of random mutations, these good mutations coming up. Are these mutations even good? Do you guys have any ideas? No, you don't think they're good? Yes, when we look at random mutations, we actually see a lot of issues with them. So cancer and aging are both due to an accumulation of mutations as cells divide. So the cells in our body, this is what cancer actually is when the cells in our body don't know when to stop dividing, and that's caused by a mutation. And aging has the same problem. They don't know when to start dividing, also due to mutations. So health policies seek to reduce the numbers of mutations we see in humans. And we have to remember there are over 3 billion potential point mutations. And a point mutation is when you change one of those letters, so remember our A, T, C, or G, we change one of those letters, we have 3 billion potential changes, mutations in humans. But only a small fraction of them, if they're mutated, have any noticeable effect at all. And these are often called silent mutations, where we often don't see anything happen. Mutations are skewed to a neutral value. So a neutral value meaning there's a net change of nothing. So nothing changes and we get a neutral change. So there is a change, but it doesn't matter. So we're going to look at this mutation distribution. So a distribution, the word distribution, just means how something's spread out over an area. And in this example, our area is a population of people. So this graph shows how most people think mutations are distributed. They think it's in this nice bell curve. We can watch where everything goes and just make happy measurements with it. But if you look at this graph, you can see that it was published in the Philosophical Transactions of the Royal Society B. So what does that name of that journal tell you? So we see that it's not actually published in a science journal, right? It's published in Philosophical Transactions. So this was published in the British Philosophy Journal. So it's not actually backed by scientific evidence. So do we think this curve is realistic, this nice happy bell curve of the way mutations distribute throughout a population? Not really. So when we look at actual mutation distribution, this is a very popular graph seen in population genetics when we're looking at the genetics of our whole population. It's called Chimera's distribution. And this is an actual calculation of mutations based on population size. So when we look at this graph, we see the words deleterious on the left side. This means mutations that delete information, that take information away. These are usually bad mutations. Then we see on the right side beneficial mutations. Beneficial mutations would be something that may add or delete information, but is ultimately good for us. And in the middle, we see this big area called the no selection zone. So there's a lot of things that can contribute to a no selection zone. And this is usually where changes are so subtle, they're not subject to selection. And they're calculated as a number. It's called NE, which is the number of people in a population, and that dilutes out how good a mutation can be. So the number of people you have, if you have one mutation in a billion people, it dilutes out how good that mutation can be if you tried to select for it. So this becomes unable to be selected for. Also, most mutations are recessive. So remember, you have your dominant allele and a recessive allele. So say blue eyes are recessive, brown eyes are dominant. And if blue eyes are mixed in with brown eyes, this is recessive and can't be selected for. Because remember, natural selection can't find or choose or decide what actually is selected for. So when you look at this, all our recessive genes also go in this no selection zone. And this is where most mutations happen. So this graph shows us that beneficial mutations are so rare that they're actually unselectable. Because you can see this graph tapers out in the no selection zone. And we actually have so few beneficial mutations in real life, you actually can't put them on this graph in any number we can actually see. So in real life, Mutations do not contribute to any net gain in information, which is needed for forward evolution. So you can't gain anything and evolve something at the same time. So how much is too much? So if we have deleterious mutations and they approach the rate of 1 mutation per person per generation, so 1 mutation and all of you sitting there per generation, long-term genetic deterioration would be certain. And this was said by Mueller, who's actually one of the biggest guys that looked at genetics, and he said this in the 1950s. When he was looking at genetics and how quickly a population can decline, and this was a model he developed in plants, he said that 1 mutation per person per generation would lead to long-term deterioration of our whole genome. The human mutation rate is actually 75 to 175 point mutations per person per generation. And this also has a lot of compounding issues. So we see a lot of problems with, as we discover more of the functional genome, we discover more mutations. So again, we're looking at the genome, and sometimes we don't know what genes do. Sometimes we don't know what things are actually for. And so as we figure out more of what the genome does, we realize that there are actually more mutations in the functions of those genes. We also see microsatellites. So microsatellites are DNA regions implicated in disease. So again, we see high rates of mutation are actually associated with disease. These microsatellites, they mutate 1 million times more often than the rest of our genome. And these are really bad. We try to get rid of these. So again, we see these high rates of mutation that evolution attributes to good things actually giving us diseases. Then we see macromutations. Macromutations are mostly chromosomal abnormalities. So our big books, remember, our chromosomes that are shaped like X's. These can affect up to millions of nucleotides, and these can cause incredible genetic defects. Often people aren't even born if they have these issues, but we see these things being passed along. So when we look at how much is too much within our genetic mutations, we actually see that not very much is too much, and we need to be careful when we look at these genes. So how many mutations do we have? So factors increase our actual mutation rate by over 200 to include over 208 mutations per person per generation. And remember our factors, we had learning more about our functional genome, our microsatellites, and our macro mutations. So these factors, when we thought our mutation rate was 75 to 175 mutations per person per generation, in each of us we have usually over 208 mutations. So Mueller again said that a mutation rate of over 1 would send humans into genetic decline. And a mutational meltdown is a real term that we use as a threat for all endangered species. And these are not evolving, remember. So this is when they start to have the same genes, their mutations aren't good, they start to fall apart, and they're not evolving in those situations when they're having these high levels of recessive mutations and hybridization and heritability. They're actually starting to fall apart. So does any of this real information match with upward evolution? And the answer is no. It points in the opposite direction. All of this genetic information that we've learned this whole time in modern genetics, when we start to learn more about the genome, we start to learn more about the human population, all of it starts to point away from evolution. There's also limitations to natural selection. So when we look at this, one of the best analogies for this is the princess and the pea. So remember the story of the princess and the pea. The only way to tell a true princess was if she slept on a bunch of mattresses and she could feel that one little pea at the bottom. Well, in this analogy, the princess is natural selection, who, remember, actually has an IQ of 0. But in this story, this is how people look at natural selection, is it's picking out something. So the princess is natural selection, the stack of mattresses is the genome in every person, and the pea is the mutation, that one little thing at the bottom. So this giant gap between genotypic change, which is our molecular mutations, all of the DNA inside all of our cells, and then our phenotypic change, which is what we actually look like, the selection of the whole organism, exists. So the princess has to look at the whole stack of mattresses to find that pea at the bottom. So remember, natural selection only sees our phenotypic change. It only sees all those mattresses. It cannot see an individual nucleotide, that little pea at the bottom, and it has to select the whole organism. So selection is based on phenotype, what we look like and how we survive in our environments, and not genotype, which is our individual letters. So we have this huge problem where evolutionists often look at all of this information as a pool of genotype rather than each individual animal and their phenotype. So you cannot separate the genes from one animal pool it with the genes of another animal, and start running statistics on that. But that is how most scientists do it. So to get around this, evolutionists have to assume that genetic units can sort independently. We can just start to spread it out. That populations are of infinite sizes. They don't look at how many people are in a population or how many wolves are in a population when they start looking at these genetics. They also have to assume that there's an unlimited amount of time for selection, which they do. They keep adding millions and millions of years every year. And then they have to assume that the ability to select for simultaneous traits, so more than one trait, can occur at the time. And again, this is very reproductively costly to the organism, and the cost of this would prevent it from evolving at all. So these are just a couple reasons that natural selection has difficulty in a real-world scenario of ever finding a beneficial mutation. We also see there's this logarithmic dilution at each level of expression. So we see when you're looking at just the string of nucleotides, you can find where that point mutation is. Then when you see what protein is produced, that might be a little harder to see. Then when you get to where that gene is expressed in the tissue, that's often even harder to even see a mutation. And then when you get to the organism, you probably don't see that mutation at all. And this would be the princess trying to find that P, natural selection trying to find that mutation within us. That's the same analogy. So when we're looking at our phenotype, what we actually look like compared to what our genome looks like, natural selection can't find any differences. So also remember, this princess in the P model is supposed to have written the genome, not just maintain it, not just evolve it. This is how we were supposed to start in the first place. So I don't know if you guys have heard the term fitness. So fitness, survival of the fittest, is the ability to survive the best in your environment. So fitness, in the genetic sense, would be the ability to pass on your functional genes, not passing on anything gross or bad, passing on functional genes. And fitness in humans is declining 1 to 2% per generation due to accumulating mutations. So your parents have 1%, 1 or 2% more fitness than you, your grandparents 1 to 2% more than that. So that's how this fitness is declining. And you can see in this graph, this is using a realistic human mutation rate, how this fitness, the ability to even pass on good genes, is declining. What you can see here is that this shows selection works, but it's on a very limited level. And this is a tough problem for evolution when we look at the whole genome. So when we're looking at this together, as evolution likes to do, they pool our genes together. They pool everything together. They don't separate it out based on individuals, where whole genomes have to be inherited as units. So this is showing that these genomes have to be inherited as units to even see where these genes are being passed on. So evolutionists like to pretend that single genes can be inherited in this infinite amount to an infinite amount of offspring based on a pool of unidentified genes. So we can't pool these genes together. We have to show a whole unit, our whole genome, being inherited to the next generation, to the next generation, and how much smaller that's getting and how much less functional that's getting as you go along, rather than just picking out genes that fit the theory. So the ultimate question here is, can natural selection create it all? This is the big question of the day. When we're looking at genetics, we want to see if mutations can ever provide a new species. And we run into this paradox when we're looking at where genes even came from in the first place. So in order to create a function at all, a new function, we would need to select for our first beneficial mutation. Remember, this isn't just a string of proteins that randomly happened in the ocean. Evolutionists now think that because, remember, our amino acids are held together by hydrolytic bonds, and amino acids build up our DNA, that somehow these amino acids came together on bubble rafts, and that's how we solve the problem in evolution, even though bubbles are still formed with lots of water. But when these amino acids were first coming together in the theory of abiogenesis, they say that in order to first make a selection, natural selection, on the first beneficial mutation, you would have to define that nucleotide's value at all. And this is just in relation to its neighbors. Based on this random string of letters, what do you choose as best? How can you select what is next best? And these things are always changing. Remember, there's no rules to this yet in an evolutionary theory. Everything's just happening together in this pool of water and bubbles. How do you decide what is the first beneficial thing? This is something that's never been solved, and this is a paradox that we see in evolution, where they say these beneficial mutations are the cause of evolution at all, but can never find a beginning. They can't even define what was beneficial in the first place. So we run into this problem of probability. And probability is usually based on computer models, and Dawkins, remember, he's one of our biggest evolutionists. He was designing a program, and he illustrated that genes can be designed. He didn't provide experimental evidence, so he showed that it was likely probable that genes could come about in this abiogenesis model. And he was trying to create this computer program. He designed this computer program. He input the parameters he liked the best. He thought we were in millions of years, 4.5 million years for the Earth. And he started just inputting all these parameters. And he just showed that we could design genes. We could design a program. And he didn't show that these things arose at all on their own. There's no experimental evidence. Remember, he didn't bother to do this in a lab. He just put numbers into a computer and hoped they came out the right way. So remember, this probability at this time of random letters generating code, a code to become anything, is 10 to the 40th place. So this is 10 with 40 zeros behind us. And this is the number that Dawkins got of the probability of genes ever occurring in the theory of abiogenesis on Earth. Then when we looked at how humans evolved and how this is assumed to have occurred, so human evolution was supposed to happen in a population of about 10,000 individuals. And remember, this is a common ancestor between what we see at other primates, so great apes, and humans. This is supposed to occur from a population of 10,000 individuals. So we quantify this as P equals 10,000, P representing population. Then we see that our mutation rate was really small. And this would be about 1 mutation rate in 30 million people. And we quantify this as delta for change with M, so change in mutation, 1 in 30 million people. In this population, this theoretical population, which is what evolutionists believe we came from, this is not creationist literature, this is using evolutionists' own statistics. In this population, we would have to wait 3,000 generations, which is 60,000 years, for one nucleotide to mutate. Just one of those point mutations, A, T, C, or G, we'd have to wait 3,000 generations for that. Then we'd have to wait 180,000 years for a mutation to become fixed in one individual. Fixed meaning you had a double copy that was able to be inherited to the next generation. So for any mutation to even be heritable, you'd have to wait 180,000 years. Then in this population, we would have to wait 18 million years for mutations to fix in the population. And fixed meaning it would be in about half the population and able to be inherited. This would be 18 million years. Then, remember, we're supposed to have evolved from apes in 6 million years. But we statistically couldn't have even have one mutation in that time. So we're supposed to be 6 million years separated from apes. But when we look at the math, when we're looking at the math that evolutionists themselves have put together, we'd have to wait 18 million years for one mutation. There's no way we could have come from apes in that time frame. Then, even further, we have to assume this process is linear, so it happened in a particular order, in a particular time frame, all at the same time, and was sequential, so that these things did not have big gaps in between their heritability. It did not change a whole lot, but we were able to do this within this very nice, happy graph of time frame. If we assume that this is actually the case, within 18 million years, we need 100 or 1,000 nucleotides. That's the average of what you see in a gene is 1,000 nucleotides. So in 18 million years times our 1,000 nucleotides, we need 18 billion years to create a gene. And this is more time than has passed since the Big Bang to create one gene in an evolutionary time frame. And remember, a recessive beneficial mutation that would increase fitness by 1% would require at least 100,000 generations, which is 2 million years, to reach fixation. So these numbers show us these are real genetic numbers. These are represented by actual population genetics. These show us that our evolution from chimps is not at all possible in the time frame provided by us by modern evolution. We would need 18 billion years to get one gene. So, moving forward, how does genetics actually represent itself in the Bible? So we've seen that with an evolutionary time frame, we can't get any new genes. But when we're looking at the Bible, you have to look at where genetics actually fit, because we're trying to represent our own perspective. So the age of a species and the age of an individual are correlated with an accumulation of mutations. The Bible details a small time when people had incredibly long lives. They often lived over 900 years at the beginning of the Bible. And then now humans have a maximal lifespan of 120 years. And we see this in the Bible in Genesis 6:3 when God actually puts a cap on the human lifespan. So if you guys want to check that out, it's a really interesting verse. We know that chimps actually have less than half of the average human lifespan. They usually live about 40 to 50 years. And then in both of these species, mutations are implicated in aging. So remember what I said about when cells start to divide too much, they start to accumulate mutations and stop knowing when to divide. And that's when we start to age. So this is implicated in all species actually around the world. So if there were initially no mutations, remember at the beginning of the world when God declared everything very good, if we had no mutations, we would expect human lifespans to be much longer. Things would know when to divide. They would know not to gain diseases. Cancer, remember, is when cells don't know when to stop dividing. They gain mutations that tell them to divide all the time. If there were no mutations when God told the world it was very good, we would expect these very long lifespans. So when we look at the lifespans in the flood, this is a graph that was done by the Bible study. And this is lifespans before and after the flood. And this is a really big spot because remember Genesis 6:3, which is right when the flood is happening, God puts that cap on human lifespan. We can see that human lifespans were really long at the beginning of the Bible, 900 plus years. And then we had this sharp decline after the flood. And we see this curve, it's a very classic curve, it's called a decay curve. And it's when we start to see things start to slope to the right like that, in that very gentle slope. And it's when we start to see things decaying, they're falling apart. So this decay curve is actually really indicative of what happened during the flood, and this is when humans began accumulating mutations. So when we look at these factually recorded dates of when people lived, we can either assume that the authors of Genesis, Exodus, Deuteronomy, Joshua, and all the other books, they either factually recorded a decline in human lifespans, or they somehow use sophisticated mathematical modeling and knowledge of molecular biology to falsify data. Only one of these things can be true, and one of these things makes a lot more sense than the other. So looking at genetics and evolution, remember, evolution says chimps and man split from a common ancestor 3 to 13 million years ago, which this is a really big range. Remember, we're supposed to have evolved from chimps in 6 million years, but we split 3 to 13 million years ago. This is a big fudge factor. So we would expect our DNA to differ. So our DNA would look different from chimp DNA by 18 to 80 million base pairs. Again, this is a really big fudge factor, a really big gap between 18 to 80 million. This also includes 150 to 650,000 generations, and this would be 60 mutations per generation. So humans and chimps do have similar genome sizes, about 3 billion base pairs, but apes, interestingly, they actually have one more chromosome than we do. This is chromosome 2, and it's supposed to be, according to evolutionary theory, it's a fusion of genetic material. And it's somehow still the same size as all the other chromosomes, even though it's supposed to have been 2 chromosomes that mashed together, somehow magically the same size. And this is also showing us that this is not really possible. We've never seen this in real life. We've never had an observation of chromosomes doing this, even in bacteria. But this is what evolutionists expect, is that this extra chromosome was somehow a mishmash of our 2 chromosomes. Evolutionists also expect a 1 to 3% genetic difference between us and apes. But the actual difference is about 40 million base pairs. This is a 12% difference. So 40 million base pairs, 12% difference compared to our 1 to 3% difference that is expected. This actually makes chimps 88% similar to us, not the 99% a lot of people say. So a lot of people say chimps are 99% similar to how we are, what we do, all of our genes, but they're actually 88% similar. Why do you think that might be? Why would we actually expect these chimps to be 88% similar to us? And there's a lot of different reasons. Common creator, that's a really good answer. Yeah, that is an exact reason. And we move similar. Those are both excellent answers. Yeah, so when we're looking at both of those answers are actually exactly what I'm looking for. So the common creator, obviously, he put all of our letters, our books of life together. But that's a really good example of maxim with we're moving similarly. So we breathe the same air, we eat the same things, we're made of the same bones, structures, all those things. All of those things have to exist in our genetic code to metabolize the air and food we breathe, to be able to build the muscle tissue and how we move. And this is actually seen with all of these other mammals. So we actually share 97%, if you're looking at that 99% of the chimp genome back when the study was first performed. And so we actually see 97% of our DNA is shared with mice in this old study. 99% was shared with chimps. So we actually see that chimps were halfway between us and mice. So we actually see that as mammals, we share a lot of our DNA simply because we do the same things. We grow hair, we need to stay warm. Just living on this earth with all these other animals means we have to share a lot of the same DNA. So this is why we see all of these things should be similar. We should not be surprised by it when we see these high levels of similarity in our genetic code. So there's this other big term called pseudogenes. Has anyone heard that word? It's a really funny word. You have? Nice. Yeah, so pseudogenes, we'll talk about those. So pseudogenes are genes that are thought to be left over from evolution that serve no purpose in us anymore. And these are a favorite of evolutionists, and this is why I wanted to bring this up, even though a lot of evolutionists are considering that we don't have pseudogenes anymore. So when the human genome was coded in 2001, the majority of our genome was thought to be pseudogenes, a vast majority of our genome. We had no idea what it did, so we called them pseudogenes left over from evolution. Then this ENCODE project, which is actually run by another evolutionary scientist, he's found that 80% of our genome is functional. We just didn't know what the genes did yet. And then Ewan Birney, he's the director of ENCODE, he predicts that we will find the human genome is 100% functional the more we learn about genes. Yeah, junk DNA, that's exactly what pseudogenes are supposed to be. So junk DNA is just things we didn't know what they were. We didn't know the function of these genes. We just called them junk, evolutionary leftovers, but in reality, we're learning all of these things play a really important role. A lot of these things are often how we interact with our environment. So our sense of smell, sometimes just very particular senses of smell or how we smell a certain flower is encoded in one of these genes. So we thought they didn't do anything because we didn't know what they did. Exactly. Yeah, we didn't know what they did. We didn't know how to even find out what they did, so we thought they were just leftovers. So a lot of people are figuring out that these leftovers don't exist. We're just figuring out what each of these things do in our body. And often they're very small things, but we're figuring them out slowly but surely. So one of these, as an example, is vitellogenin. This is another favorite of evolutionists. This is an egg yolk gene found in humans. Obviously, we don't produce egg yolk, but it's thought to be a remnant of evolution with no purpose. And as we went and looked at it, we found that it has a completely different function in mammals. It's actually important in our bloodstream. It keeps things sticky so our blood doesn't stick to our arteries and our veins. And this actually serves a really important function in mammals. It just does something different. It's the same gene and is found in a different place, but it does a different thing. So we know it is functional, and it's in a different part of our genome, which also shows that this couldn't be evolutionarily inherited. It's in a different place. It's just the same gene. We also know that it's an organic compound that likely exists in many forms in many different animals, and it is imaginative to say it's proof of a common ancestry. It's a pretty big stretch to say, even though we have the same thing, but it's in a different place, that means we came from a common ancestor. So when we look at this biochemical evidence of function, so when we're looking at the chemicals within our biology, and we're looking at how this function works, we actually see that there's function in 80% of our genome. So if we were to say that one gene in the genome that is similar is proof of a common ancestor, it's like saying the letter E in the Bible and the origin of species existing at the same place as evidence of a common ancestor. They serve completely different functions in both of those areas. And thank you, guys. So when we look at modern studies in the Bible, so modern studies are actually one of my favorite things. This is one of my favorite sciences to look at. So there's this type of DNA in our bodies. It's called mitochondrial DNA. And mitochondrial DNA is really important because it can only be maternally inherited. So when we're looking really far back in our biology, when we're first developing and we came as an egg cell, there was mitochondria in that. And because that cell is bigger than our paternal cell, what our parents or our dads give us to make us, then that's where we get that mitochondria. So because it's in a bigger cell, which is produced by the mother, this is where we get our mitochondrial DNA and why it only comes from our mothers. So what's really interesting is that the mutation rates in our mitochondrial DNA, it can be used just like a clock. So we can measure the number of mutations present in this closed genetic line to determine whether humans originated a long time ago. And with an evolutionary theory, this is about 470 mutations for evolution's 180,000 years, or recently, which is our 80 mutations for post-flood 4,500 years. And we'll talk about why we're looking at post-flood numbers. Then we'll compare these estimates to the actual mitochondrial DNA mutations seen in real humans. So this study, this used African populations, which again are supposed to have evolved first. Remember the out of Africa theory, humans came out of Africa. So this study is looking at African populations, which are supposed to be the oldest populations, and this gave evolutionists a head start. And we looked at their mitochondrial DNA. And what we actually found was that there was a mean of 78 differences in human mitochondrial DNA. Remember, an average of 80 mutations or differences in a post-flood world compared to 470 in an evolutionary world. We got an average of 78. And when you're looking at this graph, you can see where those averages kind of come from. So there's these really long, this is a web graph. So there's these really long stretches of where those mutations are, going back to 3 blue points. So we're looking at these lineages. These 3 blue points are where these differences came from. So why do you think there are only 3 points here instead of 1 tracing all the way back to Eve? This is a tricky question, and it's a really tough question. Yeah, that's a tough one. All right, so when we're looking at these 3 blue points, we're actually tracing our mitochondrial DNA, remember, to the flood, and we're tracing it to one of the 3 women that came off the ark. So remember, before the flood, there's about 1,500 years before the flood, between creation and the flood. And all of those people but 3 women died in the flood. So we lost a large population that we can't trace anymore. And that's the reason we can only trace back to these 3 points, these 3 women, Noah's daughters-in-law, that came off the ark. And each and every one of us can trace our mitochondrial DNA back to these 3 women, which is actually really fascinating. So this is why it's one of my favorite evidences in genetics that actually points back to the creation and the flood in our own genetics, the genetics inside us right now, which is really amazing. So we continue to look at this. So what happened if we were looking outside of humans? What did we see? So this study, this exact same study looking at mitochondrial DNA, was performed in 2,700 other species, and it still confirmed the creation account. So when you're looking at this graph here, you can see the evolution predicted amount of mutations, which is that big bar graph. Then you can see the creation predicted mutations, which is actually so small you can't represent it in this picture, and then the actual mutation bar graph, and one of those is much closer to the other. So they looked at these other animals, and this was only confirmation of the creation account. So each species in this picture is actually a model organism that's bred at high rates. So you can see in the middle one, our fruit fly, Drosophila melanogaster, they actually breed 10 days after being born, which is crazy. So we also see that evolution would make mitochondrial DNA really difficult to trace. So if humans evolved from a population of apes, which also contain mitochondrial DNA, a lot of things do, and we were trying to slog through human lineages and ape lineages all together, mitochondrial DNA would actually be really difficult to trace. So the ease of actually being able to trace mitochondrial DNA, which evolutionary researchers take advantage of all the time, it comes from a discrete point of origin, and that's the creation of a single woman after her partner Adam. That's the only reason we can trace any of this, is that it's actually really easy to trace because it came from one original person. So if evolution claims that rates of mutation were different in the past, then they ultimately undermine their entire position of uniformity, because evolutionists, remember, have to believe that everything has stayed the same way throughout the entire way the world's been. So we have to assume that gas has been released at the same time, mutation rates have been the same this whole time, nothing has changed. And if they say that mutation rates were different in the past in the mitochondria, then they have to undermine their entire position in order to say so. So I wanted to give you guys some practice. This is some more modern studies. So here is a journal said that evolution doesn't work quite the way they said it did. This is published very recently. So this was published in June of this year, 2018. And it was published in the journal titled Human Evolution. So this was looking at human evolution. So I want you guys to practice reading modern data and answer this question I have at the bottom, which is, why are these years different from what we see in a creation perspective? And I'll post this actual article for you guys at the end of this class if you guys want to read through all of it. I just took some snapshots to try and help you guys out. So go for it and see if you can answer this question. I know, this is a tough one, but this is getting you guys practice for the real world when you guys look at real science. Give you guys a couple more minutes just in case you guys are looking it over. So our big key here when we're looking at the study is again that this was written by evolutionists. It's published in a journal titled Human Evolution. So they're looking at these years and they're getting 100,000 to 200,000 years. So this study, again, is showing that we have, so according to a new study, people who claim that modern day animals evolved over millions of years are wrong. They're showing us that these animals came together at the same time, or came about at the same time 100 to 200,000 years ago. The reason they're getting this high number, 100 to 200,000 years, is that it's hidden in the bottom of the study, and it's included in here, but they averaged mitochondrial DNA against Y chromosomes. And this is why they did this, is they're looking at, we don't use Y chromosome DNA, we like to use mitochondrial DNA because it's more pure. Y chromosome DNA is still able to recombine with other DNA. It's able to recombine with other chromosomes. Mitochondrial DNA cannot. So they averaged, when you're looking at mitochondrial DNA, all of its mutations come from accumulating over time. So every change is an actual mutation. When you're looking at Y chromosomes, some of their changes can come together from recombining with other genetic material. That's why we don't like to use this. In the bottom of this article, you can see, I think it's the last sentence, you can see that they averaged mitochondrial DNA and Y chromosome DNA to try to get this year. And this artificially inflated the years that they're looking at. And this is why they got 100,000 to 200,000 years, rather than what creation scientists usually believe in the age of the Earth to be 6,000 years. So it's tricky that they average this and it's not included in the title, but it's hidden at the bottom of the data. This also importantly points out that all animals and humans arose at the same time. So this is, again, published in a title, or in a journal titled Human Evolution. These evolutionists are admitting that animals and humans all arose at the same time. That's a huge thing. And then in this last picture, we also see that they attribute all of this to a single sequence, a single man and a single woman, not a population of humans or a population of apes or rising from each other. They say it right here, that all of these animals and humans arose at the same time from a single sequence. So we'll see what evolutionists do with this data. Again, this was just published in June. We'll see if they come up with anything to try and explain this. But this is what the evidence is showing, even in an evolutionary, incredibly biased study. They're showing us that animals and humans arose at the same time from single sequences. All right, I have one more practice for you guys. So here's another journal article, and this is Ancient Clan Wars explains the genetic diversity drop. So what they're showing here is there was this drop in Y chromosomes. So remember, chromosomes that only belong to men and dads. This drop occurred a couple thousand years ago. So I want you guys to look at this and see what do you see from these results under a creation model? So again, this is under an evolutionary model. Evolutionary scientists did this study. What do you see in these results in this journal article? Yeah, so maximum Y chromosomes changed, but the mitochondrial DNA kept on the same path it was going. Yeah, that's a good answer. Yeah, so that's kind of exactly what you're seeing here. So when we look at this article, and again, this is an article I'll post for you guys so you can read through the whole thing and know I'm not just picking parts out to try and prove my point. This is all a huge article. I just tried to fit it all on this page. But we can see that the date range of 5,000 to 7,000 years, it fits our creation model. And then we see that mitochondrial DNA diversity continued to grow, while Y chromosomes dropped because the only men to survive the flood were Noah's sons. So remember, we had these three lineages of women survive, and then Noah's sons carried their mother's mitochondrial DNA, so we had her DNA in them as well. So we see these three lineages, but then we see that the only men that survived were from Noah's family. So we see that there was this drop, even in that generation, in Y chromosome diversity. So this is actually not a crash in the male population, but a crash in the worldwide population, leading to a decrease in male genetics over female genetics. And then this computer modeling, again, implies design, and usually designed under an evolutionary worldview. But studies like this which are forced into an evolutionary model, they still point to a creation model. So even when we're looking at this, we can still see evidence of the creation account in this force-designed evolutionary model. We can still see it here, which is really interesting. And then I have this last one for you guys. This is just another practice. This is looking at aging. So what do evolutionary scientists really think about aging? And how does that apply to what we learned about aging today with the mutation rates and the cap that God placed? And again, I'll post this article for you guys so you can read through the whole thing. Okay, so when we look at this article, we see that evolutionists, they don't have any idea how we age. They don't know if there's a limit to a human lifespan. The oldest known person that's not recorded in the Bible is 117 years old. So again, God's cap on our lifespan was 120 years. The oldest known person in modern time is 117. So we still can't meet that cap. This fits within the biblical limit. And then we see that the risk of death increases with age. And again, this is due to the mutation rate. So modern data all fits with the Bible. And this is what I really wanted to show you guys today, is that when we look at this modern data, and you guys can interpret this data too, you can look at how the creation model fits with it, what the Bible really says about what we're looking at, and see how this modern data really points towards the Bible. So, in summary, it's got a little cut up too, but in summary, we can see that natural selection is blind and purposeless, and it cannot create anything. So remember our princess and the pea analogy. Natural selection is a princess. It can't find that pea very easily. It's blind and purposeless. Mutations do not distribute evenly. Remember our chimera's distribution was not in that nice happy bell graph. It's pretty ugly. But in a way where beneficial mutations cannot be selected for. Then problems for evolution come from examining the whole genome rather than picking out your favorite individual genes like most geneticists have done in the past. You have to look at the whole genome and a realistic model in order to see what these problems in evolution are. And we have to remember it would take 18 billion years to evolve one gene in apes compared to the 6 million years to get where we are now. And this is one of my favorite quotes. This is by Nathaniel T. Jenkins and one of his cohorts, Dr. Tompkins. I cited a lot of their research here today. These are the guys that did the mitochondrial DNA study. And he said that if evolutionists want to be taken seriously in the origins debate, then they need to do more than make an isolated claim about an obscure species here and there that shows nothing but shifts in existing genetic variation or an isolated benefit due to the loss of genetic information. So that is my last slide for you guys, but if you guys have any questions, go ahead. I see one, Slutins. Why do most people still believe in evolution? I can't tell you why. I think it's easy. It's an easy thing to believe when you're not accountable to a higher power than you. And I think one of the biggest things I've heard is that evolution is the modern paganism, because you can be your own God. You can decide what you want to do, and every other person is like that too. And that's really all it is. It's a modern version of paganism where you can do whatever you want to do. Oh, you saw Nathaniel T. Jenkins at a talk? That's awesome, Luke. Evolutionists keep contradicting themselves. I think a lot of the issue is they don't know they do it. It's so widely accepted the way that they think that they don't think about what should be common knowledge and what is not. And then a lot of what they do, remember, we'll talk about this again later in the lesson, but when you look at when evolutionists say something evolved, 6 months later they might say it evolved at a different time. So their research is actually never up to date according to their own interpretations of the world. And that makes it really hard for them to not contradict themselves. And oftentimes they know they do. They just don't have a reason to try to explain it because they think most people believe the same things. These are all really good questions, you guys. I see a couple other people typing. You like the mitochondrial DNA pathway? I do too. I think it's really cool. And it's one of my favorite evidences too. And the more we learn about it, the more excited I get about it. I see Maxim typing. Why do they use Y chromosome instead of mitochondrial DNA? I think they're trying to make it fair, is why they did that. I think they're trying to make it fair to show where paternal and male influence comes from, because you guys do have your own genetic set compared to females. but it was a flawed system. It was trying to be equal where it wasn't scientific. And so they were trying to use this Y chromosome, and oftentimes in the past, before we discovered mitochondrial DNA, Y chromosomes were a really good way to trace populations because we could see these changes. Then we realized they were still really polluted. It was really hard to work through it. And we found mitochondrial DNA, which is a pure line. And so I think it was just them trying to be equal, and it was misguided. It wasn't scientific. I see a lot of you guys typing. I see Sean's, we can't use Y because of the mutation and recombination. Exactly. So the recombination makes it not pure. So it's getting all caught up in this other DNA. So you have one of your X-shaped chromosomes and another of your X-shaped chromosomes. And the Y chromosome is a little X-shaped chromosome. But it actually connects with the bigger one, and it starts to take some of its genes. And so it actually is a contaminated line. We can't trace every mutation as due to time. We can only trace it as somewhat being part of a genome and also to time. Where mitochondrial DNA, it doesn't touch other genes. It's all held within that little organelle, your mitochondria, and that's why it's pure, because every mutation we see there can only come from time. It can't be contaminated by other DNA. Maxim says they chose equality over facts. Okay, yep, that's exactly it. So the mitochondrial DNA doesn't change when we combine, where Y chromosome does. Yeah, exactly. So again, the mitochondrial DNA is held in that nice package, that organelle, that's only passed on through the mom. And the Y chromosome is not packaged the same way. It touches all of our other 23 chromosomes. It touches everything else. So it can recombine with all kinds of other genes that come from our lineage, our other generations of ancestors. So it's not a pure line. See a couple people typing. You guys are asking really good questions. These are great. The next class is at the same time next Thursday, and this will be recorded for you, William, so you guys can check this out afterwards. I just won't be available for questions, but if you have more questions, just save them for next time. But thank you for coming. Yeah, of course, no problem. Why does mitochondrial DNA not change or recombine? Yeah, so mitochondrial DNA, remember, so we have all these organelles in our cells, and our mitochondria is one of them. And this is actually held in a double envelope, so it actually has two layers of, it's not real cell tissue, it's these lipid bilayers. So it's just a bunch of chemicals that keep it packaged together. And so what actually happens is all of that DNA inside of it, it doesn't touch anything else. It's just held in that mitochondria. Evolutionists actually think that way back when we were supposed to be bacteria, that the mitochondria was separate bacteria and it got engulfed by another one, and then that was somehow magically inheritable. That's never been observed, but they thought that we passed it on somehow. But because it's held together in the envelope, it doesn't touch the rest of our genome. So it's just passed on as the cells divide. It's always that exact same sequence. It never has a chance to change unless it mutates. So that's why it looks like that. Yeah, of course, Sandra, no problem. See a couple other questions coming up. Is the study of genomes, DNA, etc. called molecular biology? Oh yeah, so there's a lot of different questions. So yeah, molecular biology is the big name for all of that when you're looking at genes. There's also genetics, which is looking at the gene itself. So remember just when we're looking at it, just the words. When we're talking about the genome as books, those are the words. Then we have genomics, which is looking at all of our DNA in one person. There's population genetics, which we talked about a lot today, which is looking at the genes spread out along a population. And then you can also look and see what genes do later on. So there's RNA and proteins that genes turn into that transcribe things and keep us moving. And so you can look at transcriptomics and proteomics. There's actually a lot of things, if you're interested in genetics, to look into, which is really exciting. Yeah, no problem, Caitlin. This is really fun. I'm glad you came. The next topic is flood geology. So we're going to take this another step, and we're going to look at how the flood actually shaped our Earth. And we're going to look at fossil evidence, climate evidence, and geologic evidence. What could have the Nephilim done to the gene pool? That's a good question. And it's really hard to trace because, again, that was before the flood. So remember, the Nephilim were a very small population. They're only mentioned in one verse in the Bible. And that was before the flood happened. So there's actually not any way of knowing because we lost so much population and genetic evidence prior to that flood. and we had our genes kind of bottleneck. So that's when we had a big population first, then a little population, and now we're back at this big population. And past that bottleneck, we can't really look because we've lost so much evidence there. So that's a really good question. It's just there's no way of telling where we are now. How many genes are predicted to be pseudogenes? Right now, 20% of our population is predicted to be pseudogenes. But again, the founder of ENCODE said he doesn't think anything is actually pseudogenes. So the more we learn, the less pseudogenes we find, and we replace them with actual genes. That's a very good question. Oh yeah, perfect. Sean just put when the next lesson is. So we'll be doing that next week. So I hope to see you guys all there. Perfect. Yeah, so again, this will be a 6-week lesson. We'll be doing different topics every time. So keep coming. It is totally okay if you guys ask questions from the previous lesson at the end of the next one. And again, we have 6 of these, so you have 6 chances to ask your biggest questions. And I'll keep giving you more of this awesome information and more modern information. Maxim says, why do Y chromosomes not have the same envelope? So remember, these are in a nuclear envelope. So when we're looking at all of our DNA, it's within the nucleus of all of our cells, and our mitochondria is over here in a different thing, in a different envelope. So the rest of our DNA, including our Y chromosome, is all in that nuclear envelope, and that's 23 whole chromosomes. So it's touching each other, recombining, doing everything it needs to do to keep us diverse and healthy, where our mitochondria stays over here and never touches it. Yeah, of course, I'm glad I could share. Thank you for coming, Clara. Do you guys have any other questions? Okay, see Maxim. So as a boy, does your maternal mitochondrial DNA not get passed on to my children? That's exactly what that means. So you inherited that from your mom, and there's nothing else. Everything's come from your grandma, the woman before that, etc., etc. So your mitochondrial DNA from your mom will never be passed on to your children as a boy. But you always have the chance to pass on your Y chromosome DNA, and that's still unique to you. It is interesting. Yeah, I think it's one of the really cool things about when you look at cellular biology and how everything kind of pieces together. It's really interesting how we're put together. Yeah, thank you so much. I really enjoyed it. I think we're done with questions. Okay. Yeah, if anyone else has any other questions, you guys can hang on to them for next week. We'll be here at the same time next week. Okay. See you